Hi everyone. I welcome all of you to yet another Fusion Talk from our Fusion Talk series. So before I go into, I'll first tell you what Prescience is. So Prescience is the company that is founded by academicians from IIT and NCL. At Prescience, we team up with both academic and industry leaders to address challenges in drug and material discovery. And we take immense pride in being one of the first companies in India to develop our own in-house drug discovery platform, well equipped with advanced AI features. With that, our mission is to make drug and material discovery accessible to everyone, revolutionizing the scientific landscape and paving the way for groundbreaking innovations. Regarding Fusion Talk series, so this series have came from our wish to get academic and industry experts to share their valuable insights into advancing scientific field. Now about speaker, that is Professor Ashish Kulkarni. He's an associate professor of chemical engineering at UMass Amherst. He was an instructor of medicine at Harvard Medical School and associate bioengineer at Brigham and Women's Hospital. He has done his BTech from ICT India and PhD from University of Cincinnati and postdoctoral training from Harvard Medical School, MIT. His lab essentially works on immuno engineering, bridging nanoscience drug discovery and engineering design with manipulating the immune system to address fundamental and translational questions in cancer. And working in this field, he has published several articles in top tier journals. And we'll be looking some of his work with the title of his talk, that is Nanoscale Approaches for Therapeutic Immune Modulation. And now I would ask Professor Kulkarni to start his talk, and we will be looking forward to the exciting talk.
Okay, thank you, Anil, for the kind introduction, and thank you, Seema and Sudeep, for the invitation. I'm really excited to share our work here today. First, I would like to begin by thanking my outstanding students who have done amazing work that I, take credit and share the work. Although I love all of their work, today I'll be only focusing on the work done by Anujan, An, and some of my initial work as well. But again, I would like to thank my postdoctoral students, graduate students, undergraduate students, and collaborators who have worked with us over the last 7.5 years, and the funding agencies. So our lab works on three main platform technologies. As Anil mentioned, we work on immunoengineering, which at the interface of engineering and drug delivery with immunology. We have three main platform technologies that we work on. The first one is to design microscale platforms that can allow us to understand tumor cell immune cell interactions, allow us to identify novel targets, screen drugs, and so on and so forth. We also design drug delivery systems that can allow us to deliver drug to different immune cells in the tumor microenvironment with the hope that that will lead to better anti-tumor effect. And we also work on designing imaging nanoprobes that can allow us to monitor immunotherapy responses early on. Now, as I understand that we have a very broad audience with diverse expertise and understanding. So let me start by, giving you a brief overview of what our immune system is, and then I'll talk about how tumors overcome immune responses and what are we doing to make sure we can activate those immune system to target tumor cells. So our immune system is classified into two types: innate immune system and adaptive immune systems. And broadly speaking, there are multiple cell types that are involved in responses to each immune system. Now, the innate immune system, which is a very primitive first line of defense, which has, again, multiple immune cells that are responsible. This immune response is rapid and non-specific. Whenever you are exposed to any foreign bodies, this could be pathogens, viruses, or bacteria. The job of these immune cells, innate immune cells, such as macrophage or antigen-presenting cells, is to take up these foreign bodies, degrade them, and eliminate them. So they have same type of responses, broadly speaking. They're rapid and non-specific. And NK cells can go and target the infected cells. Now, our innate immune cells, as I said, is very rapid response. It can happen within few seconds to a minute, and they're very, very effective in clearing up foreign bodies that you're exposed to. But a lot of times what happens, even though this is an effective response and clear up the foreign bodies, most of the time, a lot of times what happens is that these foreign bodies are clever. They can overwhelm the innate immune system and start growing. At that point, the second type of immune system is called into help. So these are adaptive immune cells. Now, adaptive immune cells need to be activated. They don't really have same type of responses, right? These are adaptive, as name suggests. They adapt to the type of infection, type of pathogens or viruses that they are exposed to. They need to be activated, and that activation happens through innate immune cells, such as antigen-presenting cells, that can present specific markers on T cells or B cells, and then they can go and target the infected cells or pathogens. So T cells can go and recognize those markers on infected cells and kill them, and B cells can release antibodies that can go and target the viruses and eliminate them. So we have innate and adaptive immune system, right? And then they're very effective in different types of responses. They have broad types of responses. So even though you are exposed to, once they're educated to particular viruses and pathogens, you are protected, right? So that's why when you take vaccine, you are protected from that type of pathogens or viruses for your life. And your immune system is so effective that even though you are exposed to millions of bacteria and viruses every day, you don't feel anything because your immune system is working to clear them up around the clock. Now, now this is good because now we are thinking about foreign bodies which are not part of your body. So your immune system is very effective in clearing them, recognizing them as because they're foreign bodies. Now the question that comes is for cancer, which is not a foreign, right? These are cells from your body. Does our immune system recognize cancer cells which are part of your body? And the answer to that question is yes. Our immune system is capable of recognizing and eliminating cancer cells as well. So let me show you two videos to demonstrate that our immune system is capable of doing that. So in the first video, I'm showing here, as I mentioned, adaptive immune cells such as T cell is kind of seeking out, binding, and killing the cancer cell, right? That's the video it shows here. And so our adaptive immune system is capable of recognizing and targeting your cancer cells. In the second video here, I'm showing an innate immune cell and macrophage that can eat up, degrade, and chew up cancer cells, right? So that's their job. Innate immune system or innate immune cells can actually take up your cancer cells and degrade them. So just to show you a couple of examples, obviously there are multiple pathways, multiple cells, multiple ways that immune system can recognize cancer cells and target them and degrade them, right? So, we know that immune system is capable of targeting cancer. The question that comes is, and that has been, the question of the research for a lot of time is, how do tumor cells grow despite this effective and very, very good immune system? The answer to that is cancer is smarter, right? They have developed various ways. Cancer uses every trick in the book to evade immune responses. For example, cancer can recruit these immune suppressive cells, such as regulatory cells that can dampen the immune responses. They can release immune suppressive factors that name suggests. These are immune suppressive. Their job is to suppress the immune system, right? They can release these immune suppressive factors, which are proteins that can modulate the functions of immune cells, such as tumor-associated macrophages, which are supposed to go and eat up tumor cells. They will help tumor grow. Tumors can also overexpress proteins that can bind to T cells, right, which is an adaptive immune cell that can actually, create a break or acts as a break to the tumor cells. In other words, these activated T cells that are ready to go and kill the tumor cells, when they bind to this ligand, they will stop, right? They will not kill the tumor cells. And tumor cells have developed these ways of overcoming responses by both innate and adaptive immune system. Now, there have been a lot of research that has been gone on to understand these mechanisms and perturb these mechanisms. One of the best example is antibodies that can block these interactions, which are called immune checkpoint antibodies. And understanding that if you block these interactions effectively, you can unleash the T cells that can clear up tumor cells. And this has led to a breakthrough therapies. Some of those are now FDA approved, and kind of gave a way of targeting these aggressive tumors. For example, patients who were given few months to live are now surviving for decades, right? So this has had a tremendous impact on a lot of aggressive tumors that are very advanced tumors that didn't really have any options. So understanding these mechanisms uh, have led to completely changing the landscape of how patients are treated, especially for advanced cancers. But despite this initial excitement, there are several challenges that have been observed, and those are the things that we are interested in working on. So one of the challenge is the low response rate. As you can see, some of the blockbuster immune checkpoint inhibitors have around 20% or so response rate as a monotherapy, right? So 1 in 5 patient responds to these treatments, where these are obviously very expensive treatments. They have high toxicity. You can increase this by adding combinations, but that also adds toxicities. And one of the problem is, if you are patient who are going to doctor and getting these expensive treatments without a promise of whether this will work or not, you don't, as a cancer patient, they don't really have time to do these therapies that may or may not work. So the idea was, can we actually go from here to get responses all the way, as many patients can respond to these treatments from as many tumor types possible. That was the first question that we wanted to think about. And the second question was, can we actually stratify early on patients that are responding from patients that are not responding? In other words, can we actually design imaging probes that can allow us to monitor if the patient are going to respond or responding or not early on, and then change the treatment before it is too late? So the two main important questions that we have been interested in answering is, can we design nanoscale approaches that can allow us to modulate immune responses? We are morely focusing on innate immune responses because there's a lot of work that has gone on to done in adaptive immune responses, but we are focusing more on innate immune responses that can be used to generate anti-tumor immunity, and that can lead to tumor regression. And the second question is, can we design nano reporters, as name suggests, can they report back, or can they let us know if the tumors are responding or not in real time, right, at early on, so that not only understand whether those tumors are responding, but can also differentiate between responsive and non-responsive tumors, so we can kind of make early decision whether we need to add therapies, we need to change the treatment, or do combinations. So there are two main questions that we are trying to address. So I'll focus on the first question first. We although worked on a lot of different types of cell type, innate immune cells in the tumor microenvironment. Today I'm going to focus on macrophages. Now macrophages are this, innate immune cells. As I said, their job is to go and eat up tumor cells, but they're also very fascinating cells in terms of inflammation as well. So it is shown that in tumors, there's a high proportion of these macrophages that are infiltrated, especially in solid tumors. There's a huge mass of macrophages that that are infiltrated in the tumor. Now, they are there. The higher percentage of these macrophages has been shown are correlated with poor prognosis, which means that the more number of these macrophages are there in the tumor, that for a lot of solid tumor types, that can lead to poor prognosis. The tumors don't respond to treatments. They can have a chemotherapy resistance, and so on and so forth. So our idea was, can we actually think about targeting macrophages that can lead to anti-tumor responses? Because they are in high number in the tumor microenvironment, they're helping tumor grow. Can we actually target them? So before we go on to talk about what we worked on, let me just give a brief background about what macrophages do in the tumor microenvironment and what we are planning to do. So macrophages are these fascinating cells because they can actually shuttle between different phenotypes based on the environment they're exposed to. And they're generally in a continuum of different phenotypic states, and there's a spectrum of these phenotypic states, and the two end of those spectrum are M1 and M2 like macrophages. Now, M1 like macrophages are more inflammatory macrophages. they are have anti-tumor functions. Their job is to activate immune responses and reduce tumor growth, right? So ideally, we want higher M1 macrophages in the tumor microenvironment, whereas M2 macrophages are have pro-tumorigenic functions, right? They lead to tumor promotion, metastasis, which is growing of tumor in distant organs. They can lead to impairment of adaptive immune responses such as T cell responses. So, we don't want higher M2, we want higher M1 in the tumor microenvironment. But as you can imagine, when the tumor starts growing, they have predominantly higher M2 macrophages, which helps tumor grow. So how do they get into tumor? Generally, the circulating precursor cell called monocytes are recruited to the tumor microenvironment by these chemoattractants, and they can then differentiate into either M1 or M2 like phenotype based on the environment they're exposed to, cytokines they're exposed to. Now, for advanced tumors, most of them, start secreting, as I said, immune suppressive cytokines or immune suppressive proteins that can lead to more M2 like macrophage phenotypes. And you have this, CSF1, which is a coronally stimulating factor 1, which is a cytokine that is important for M2 macrophages. So tumors can release large number of this CSF1, so add M2 macrophages as well, that can lead to more and then that can create an environment that is, sustaining more M2 macrophages. So then, this has been known. So a lot of strategies have been developed. For example, strategies that can prevent the recruitment of monocytes to the tumor microenvironment, strategies that can prevent the differentiation of this monocyte to M2 macrophages by targeting several cytokines, or depleting M2 macrophages altogether, right? So just, during the therapies that can deplete M2 macrophages in the tumor microenvironment. But what has been shown that if instead of, using these treatments, which, once you stop these treatments, there's a rebound increase that happens. If you can reprogram them from M2 to M1 phenotype, you can get much better effect, because now you have higher M1 that is endowed with these anti-tumor functions that can lead to better immune stimulation, tumor suppression, and long-term survival. So one of the question was, can we actually reprogram them from M2 to M1 phenotype, right? So higher M1, lower M2. So this was one way, but then we also want to think about another challenge. As I mentioned, tumors can use every trick in the book to overcome responses. Then another trick, as I said, your macrophages, are supposed to go and eat the tumor cells, but tumor cells can overexpress something called CD47 that can bind to CYP-alpha on the tumor-associated macrophages and acts as don't eat me signal, right? So basically, instead of them eating up, chewing, and degrading tumor cells, this acts as don't eat me signal. As you can imagine, most of solid tumors overexpress this CD47 that acts as don't eat me signal. So even if we reprogram macrophages from M2 to M1 phenotype, they'll go into the tumor, and then this will act as a don't eat me signal. So they won't be able to function as well as they wanted. So one of my first graduate student, Anujan, he kind of thought about a strategy where he wanted to reprogram macrophages from M2 to M1 phenotype by doing efficient this CSF1R inhibition, right? That can lead to better lower M2 phenotype and higher M1 macrophages in the tumor microenvironment, while also blocking this don't eat me signal, which is CD47 CYP-alpha signal. The idea was, if you can, reprogram them and we can block these interactions, we get macrophages that are going to be very potent, also very effective eaters of the tumor cells, right? So the idea was we can design this using a nanoparticle. Now, nanoparticle has been widely used to deliver drug to the tumor because, nanoparticles can get into the tumor at much better, much higher concentration compared to other organs if you use the right sizes, if you use the right targeting mechanism as well. So we wanted to design this nanoparticle system that can have dual blocking mechanism. One of the problem was we wanted to think about, instead of trying a lot of different types of nanoparticle systems and doing permutation, combination, empirical approach, we wanted to step back and think about, can we design a system by understanding if we can, understand how drug interacts with that nanoparticle and make the system much better so they can deliver as much drug or as much inhibitor into the tumor microenvironment as possible. So this is where we, started working thinking about computer simulations because computer simulations are amazing in terms of getting atomic details of biomolecular interactions that you cannot get with experimental techniques, right? You can get how molecules are interacting, how different interactions are happening in real time. You can get the spatial and temporal resolutions that you cannot achieve with experimental techniques. So we wanted to use these computer simulations to design our nanoparticle with high drug loading and high stability, right? Those were the criteria. So we work with Sudip. Now Sudip is here. Sudip has been outstanding, and Prithvi was a graduate student in Sudip's lab, and they have done amazing work in terms of designing computer simulations. I think this was, because these computer simulations were very common in terms of drug lipid interactions, mostly as a cell membrane mimic, but not so much in terms of delivering, not so much in terms of designing a nanoparticle system. So this was a kind of a first of its kind of algorithm that can allow us to design these nanoparticle systems. And Sudeep might be able to give you a lot more information about what goes on into running these simulations. But let me just walk you quickly through what the idea was. The first idea was to pick up a drug, a parent drug or template drug, a molecule that has some biological activity, but makes the nanoparticle system very, very unstable at a high concentration that we want to use. We then run simulations, all atomistic simulations, and the automated analysis of stability. If it's unstable, we do a structural changes in a drug, small changes in the drug, run the simulations again until we identify a drug that actually, a structure or changes in the structure of the drug that makes it stable. And then once we identify that, we chemically synthesize this molecule, and then we make the nanoparticle and do extensive biological studies. So let me give you a couple of examples to show how beautiful this platform is and how powerful this platform could be in thinking about designing systems that could lead to better drug loading and stability. So we started with paclitaxel. Paclitaxel was is a very widely used chemotherapy drug for a lot of different types of tumor, but it's a bulky drug. It's a big molecule that is very notorious to form stable nanoparticle system, right? Especially lipid-based nanoparticle system. So we started with doing that. So we loaded a high amount, around 20 mole% of this paclitaxel in the lipid bilayer. As you can see here, with the free drug, it forms these large aggregates at the interface, right? And because this is a hydrophobic drug, for a long time, people thought that it really likes to interact with the hydrophobic part. It does to a certain extent, but it also forms these large aggregates. So our idea was, can we actually overcome these drug-drug interactions, drug-drug aggregates that are happening, and see if we can stabilize this? So we conjugate that to a small lipid using a small linker, flexible linker. Again, linker and the lipid both are critical and making this stable. I'm not going to go too much detail into that, but just show you the structure that we finally identified called STX2. When we ran the simulations again at higher mole%, we see that the aggregates were reduced significantly. It forms a stable bilayer system, and we can get much stable system at much higher drug loading. Obviously, we made this experimentally and showed that it forms very stable system. I'll give you another example. Now here in paclitaxel, you had these random aggregates that were formed, which led to instability. Another type of molecule we started was PI103, which is a PI3 kinase inhibitor. Again, it was widely studied and experimentally used for not clinically, just the preclinical studies for a lot of cancer types. This is a planar molecule. It's a smaller molecule compared to paclitaxel. So our idea was, since a hydrophobic molecule, smaller molecule, it should easily go into the lipid bilayer and form a stable system. It was not the case. We actually saw that it forms very unstable nanoparticles, more unstable than paclitaxel. And when Prithvi ran the simulations, we saw that it's actually, it's very interesting the way it kind of interacts. In paclitaxel, we saw these aggregates, large number of aggregates, right, that are random aggregates. In PI103 with PI3 kinase inhibitor, it forms these pi-pi stacking interactions because this is a planar molecule, right? And because of those pi-pi stacking interactions I can see here as well as here, it forms a lipid bilayer that is very unstable. Same way when we identified in a molecule, and then did the simulations, it forms a stable bilayer system. You don't see any pi-pi stacking, so we prevent these interactions. By preventing these drug-drug interactions, we form a very stable molecule. So in other words, this system can not only give you whether you have stability or not stability, it can also tell you what type of interactions are happening, and then if there are ways to overcome those interactions. In this case, these are drug-drug interactions. There could be drug-lipid interactions. There could be other types of interactions that can make this system unstable. But understanding that can give you a better idea about how to design a better nanoparticle drug delivery system, right? And that will be a very powerful way to make sure that you design the right way and make the molecule right way that have better chances of becoming stable as well as higher drug loading. So we use this platform for a lot of different types of drugs. We used it for anywhere from chemotherapy to immunomodulatory drug to kinase inhibitor, epigenetic drugs as well. As you can see here, going from template drug, which all you see is having this large aggregates, that formed in a tube, to very stable system. Again, you see there's a rupture that happens with the bilayer, whereas it forms a stable lipid bilayer with 6 to 10-fold higher drug loading. So this is a very powerful platform that can be used for rational design of nanoparticles drug delivery systems. You can use any drug or lipid molecule. We use the lipid excipients that were available, but you can potentially use any kind of systems to design this computational algorithm. Now, let me go back to the macrophages. I digress a little bit, but this is something that I really find interesting, that how we can use computational systems to understand mechanisms and design more powerful drug delivery systems. So for the macrophage reprogramming, we used started with same way BLG 945, which is a commonly used CSF1R inhibitor. It's very potent, but it's very notorious in terms of efficacy. It only works for a few minutes. It gets cleared up from the circulation once you inject very quickly, which means that you have to give very high doses of these drugs without significant effect. So when we ran the simulations, it's same way. We had stable the aggregate that formed at the interface, but when we identified the structures and ran the simulations, it formed a very stable lipid bilayer system. Same is the case, same as what we showed it previously. Then, once we had this identified molecule, we made the nanoparticles. We did a structural characterization of this to show that it forms nanoparticles around 100 to 150 nanometer. They are stable during storage conditions. This is 4 degrees, so they are stable for over a month, which means that we have higher drug loading. We also have higher stability. But it can also release drug in the cell lysate, which is what we want, right? Once the nanoparticle goes inside the cell, we want the drug to be released in a sustained manner, right? Sustained manner is very important because we want the cell, the signaling to be inhibited in a sustained manner. We can also get drug, not only get drug inside the cell using a nanoparticle. So nanoparticle is getting similar drug at earlier time point, but the nanoparticle retains the drug inside the cell for longer period of time, right? Where the free drug gets cleared up very quickly. So only traces amount of this drug was observed at 18 hours. So, in summary, what we have is optimum size, right? Stability, sustained release, and higher drug accumulation and residence inside the cell for a longer period of time, right? So this is not killing the cells, but keeping the concentration just high enough to get better inhibition. So we wanted to kind of see, okay, do you really get the inhibition of the pathway that we needed? So we ran Western blot to see how much inhibition do we get. And you can see here we get inhibition using nanoparticle for as long as 72 hours, whereas the free drug only works for 5 minutes. After that, it stops working. And when we actually quantified for multiple time points, we see that we get the nanoparticle that inhibits for as long as 72 hours, right? So now we have a system that can actually make a stable nanoparticle that can release drug in a sustained manner, that can retain the drug in the cell for a longer period of time, but also inhibit the signaling for as long as 72 hours. So this is the first time anybody showed that we can design a nanoparticle system that can, lead to sustained inhibition of this kind of kinase signaling pathway. Now we wanted to also see, does it actually inhibit downstream signaling pathways as well? So we did a lot of different pathways. I don't want to go too much detail into that. All I want to show here is that we inhibit the signaling, both CSF1R and downstream signaling pathways that are, we inhibit the signaling that is important for M2, and we activate the signaling that is important for M1. So at least at protein level, we see that we are going towards getting more M1 by having this sustained inhibition and lower M2. The idea was, can we actually do now surface marker analysis? So we did flow cytometry. It's an technique that is used to figure out what are the surface markers that are expressed, or surface proteins that are expressed on this M1 versus M2 macrophages. So both M1 and M2 macrophages have different types of surface markers that are expressed, and you can use those to differentiate between how much percentage of M1 versus M2 do you have. So CD206 is an M2 macrophage marker, and MSC2 and CD80, and we also did CD86, which are M1 markers. So ideally, if your system is working, you're inhibiting the pathway, you should see lower CD206 and higher MSC2 and CD80, which is what we see with the nanoparticle. These are two different time points, 24 and 48 hours. We do see that we get much lower CD206 and much higher MSC2 and MSC1 when you use a nanoparticle compared to free drug. And we did this with over different time points. As you can see here, the free drug doesn't work at all, but the nanoparticle can show much higher M1 to M2 ratio for as long as 72 hours. So this was very exciting, because now we have a system that can, not only inhibit the signaling, but reprogram macrophages from M2 to M1 phenotype very, very efficiently, much better than the free drug. Now we wanted to see, okay, next step, okay, now we can get this all in vitro in cell-based assays. What about in tumor models? Do you actually get more drug in the tumor? We did that. We actually saw more than 8-fold more higher drug accumulation in the tumor when you use a nanoparticle compared to free drug, and that translates into significant anti-tumor efficacy, right? So we have seen here that we get much better anti-tumor effect using in melanoma as well as metastatic breast cancer, right? We get much better effect using a nanoparticle. And I want to remind here that we are not targeting cancer cells, right? We are targeting macrophages in the tumor microenvironment that are getting reprogrammed and targeting tumor cells. And that is leading to significant anti-tumor effect without as much toxicity. And as expected, we get much higher M1 to M2, and that can lead to better survival as well. Now, that was the first question, right? We design a system that can reprogram from M2 to M1. We still need to figure out blocking the signaling so they can get much better effect at maybe less number of doses, or less, number of less amount of drug that we need to inject. So we wanted to think about, can we block this interaction using an antibody on the SIRP-alpha, because we wanted to target macrophages, not tumor cells. We wanted to think about targeting macrophages by using this antibody on the tumor-associated macrophage. So we, Anujan designed this nanoparticle system that had, anti-SIRP-alpha antibody conjugated on the nanoparticle, the nanoparticle that I showed previously. The idea was this can block SIRP-alpha CD47 interactions that can lead to better phagocytosis, while also reprogramming macrophages from M2 to M1. Now, in this case, we also saw that having this SIRP-alpha antibody on the nanoparticle acts as a better targeting agent. In other words, the antibody targeted nanoparticles, we get much higher accumulation in the tumor compared to non-targeted ones, right? So this can also act as targeting in addition to blocking these SIRP-alpha CD47 interactions. And if you remember in the previous studies, we did 3 doses. Here, even a single dose of this combination therapy led to much better anti-tumor effect, right? So you can see that if you do a rational combination that can have, that can inhibit the signaling much more efficient way, you can get better anti-tumor effect at much lower doses. So we explored this further, and Anujan and several other students in the lab have performed combinations, single therapies for a lot of different types of targets in macrophages to see how much reprogramming can you get from M2 to M1, and identify several potential pathways that can be used as well. Okay. So to just summarize this part of talk, I've hopefully convinced you that we can design nanoscale approaches that can target innate immune cells in the tumor microenvironment, such as macrophages, that can lead to significant anti-tumor immune response and and lead to tumor regression. Now, in next few minutes, 10 minutes or so, let me talk about nanoreporters, right? Designing imaging probes that can allow us to monitor immunotherapy responses. Now, the first question that you might ask is, why do you care about monitoring immunotherapy response? The answer to that is monitoring immunotherapy response is trickier than normal chemotherapy or radiation therapy. And let me tell you why that is. So for normally chemotherapy or radiation therapy that targets cancer cells, you get two types of responses broadly. Either tumors are responding where there is an initial response that happens, or tumors are not responding where disease progression happens, where tumor grows, right? So you have either yes, tumors are responding, or no, tumors are not responding. But in case of immunotherapy, there are additional patterns of responses that have been shown. For example, you see that there is a stable disease, so the tumor don't grow, they just maintain the same tumor volume. Or when there is a activation of immune system, this activated immune cells flock to the tumor, and that can lead to increase in the tumor volume, right? Because there are high number of immune cells that goes to the tumor that can easily misunderstood as disease progression, which is why it's called pseudo progression, which at the later time leads to reduction in the tumor volume. The problem is, currently there are no ways to differentiate between disease progression and pseudo progression because you're monitoring their overall tumor volume or metabolic profile. Both can be difficult to understand based on just the size measurements or metabolic changes that happens. So our idea was, can we actually design systems that can monitor these responses early on, but at the cellular level? Even our studies, we have shown that monitoring tumor volume or using metabolic changes using PET CT also is not really effective because you don't get any responses. So as I said, we wanted to kind of go into a much cellular level and kind of see if we can design systems that can monitor cellular level changes that happens in the tumor microenvironment when immune cells are working, immunotherapy is working. We can get these responses early on, much early on, before these tumor volume changes can happen. So we wanted to kind of go back to the basics and see how does tumor cells get targeted by immune cells, right? I showed a video, but here a little bit more mechanistic overview of what that means. So you have cytotoxic T cell, which is an adaptive immune cell, target tumor cell by two main ways. The first main is the granzyme B perforin pathway. Very simplistic way of showing here, but perforin is a pore-forming protein that forms the pores in the membrane of the cell, and then granzyme B can then enter into the target cell that can lead to activation of this caspases that can lead to apoptosis. And there are several other pathways that are there as well. But we are focusing more on granzyme B and caspase 3 levels. Now, these are the enzymes that are important. And the idea was, if you can monitor the activity of granzyme B or caspases in the tumor microenvironment, in the tumor cells, we can potentially say that, the T cells are killing the tumor cells and can monitor immunotherapy responses early on. In other words, we can actually use this, activity of these enzymes as a direct readout of immunotherapy responses. So my graduate student, who's now a scientist at Bristol Myers Squibb, she designed this granzyme B nanoreporter that can not only deliver immunotherapy drug, but also an activatable imaging agent. Imaging agent being granzyme B responsive linker that can capture the outcome of immunotherapy activity in real time. So the two scenarios that we could think about is, if the tumors are poorly immunogenic, there is no immune response happening, immunotherapy is not working, you will not see activation of caspase or granzyme B, your tumors won't show any signal, right? So tumors will not be showing any fluorescent signals if you're using fluorescent reporters. Whereas if there's a highly immunogenic tumor, you should see a stronger responses that are happening. So she designed this studies where she used on one side poorly responsive tumor, on another side highly responsive tumor. And the idea was, we use anti-PD-L1, which is an immune checkpoint antibody, as an immunotherapy drug. And we have a granzyme B reporting probe that will tell you whether the granzyme is activated in the tumor microenvironment or not. So ideally, poorly responsive tumors should not show as much activation, whereas highly responsive tumors should show a lot of activation. And we did 3 different doses. As you can see here, these are bright field images. These are just showing the location of the tumor, but these are fluorescent images. As you can see, the dotted oval shows the location of the tumor that is poor, that is highly responsive. So highly responsive tumor, as you do more doses, more treatments, they light up, right? So you see more responses in the highly responsive tumors compared to poorly responsive tumors, which don't show any responses. And we use the control antibody, which is a control drug, in this case, control antibody that should not activate immune responses. You don't see much activation happening in the tumor microenvironment. And you can see, we have significant difference between responsive versus highly responsive versus poorly responsive tumors that we can capture at a much earlier time point where the tumor volumes are not even that much different, right? We don't really see as much changes in the tumor volume between treatment versus control versus vehicle groups. So in other words, we can capture this immunotherapy response at much earlier time point using these activatable nanoreporters compared to tumor volume changes, right? Which is what was very important to show. And then you can basically see these are tumors. These are the poorly responsive tumors. These are highly responsive tumors. You see that these tumors light up, right? So you get much better responses that are quantified here if you use activatable systems. And obviously, we get much higher granzyme B and much higher cell death. So here we have shown that, if you design activatable systems that can monitor cellular level changes, we can get much better anti-tumor, we can monitor the responses much better, much quicker as compared to monitoring tumor volume changes. Now, around the same time when Anujan was working on this, his macrophage immunotherapy systems, he was around, the same way that, he wanted to see whether he can monitor the macrophage reprogramming that happens in real time, because all we could see was tumor volume changes that happen at later time point, not so much in real time macrophage responses. So he wanted to think about designing nanoreporters that can monitor macrophage reprogramming that happens. What he found out was, the macrophages have 2 main molecules that can be monitored that are relatively selective for macrophages. So nitric oxide is a hallmark signature for M1. That's the molecule that can go and kill the target cells. Whereas M2, we have arginase 1 that is released by M2 macrophages that help them travel and help them grow as well. So you want to think about designing imaging probes that can monitor nitric oxide, which is a small molecule, and arginase, which is an enzyme, right? So monitor these levels that can then allow him to figure out the relative M2 or M1 levels in the system or in the tumor microenvironment. So he designed this M2 nanoreporter using a dye and a quencher and a linker that is arginase cleavable. The idea was when arginase is activated, it will cleave up this linker and your dye will get activated. Same is the case with M1. He used a very specific linker, which is responsive to nitric oxide that can, once nitric oxide comes into close contact with this linker, it forms a 5-member ring and then destabilizing this bond and, again, cleaving these molecules to get a dye that gets activated. And he used this M1 and M2 nanoreporter for 3 different types of systems. I'll quickly go over those. So he used it for, so M2 nanoreporter, as I told, M2 macrophages are in high numbers in the tumor microenvironment. So if you can use M2 nanoreporter to monitor tumor progression, you can basically see where the location of the tumor is and how much tumor is growing. We can use M1 nanoreporter for local inflammation. We can also use it for monitoring immunotherapy responses, right? If your system is reprogramming M2 macrophages to an M1 phenotype or not. And we use this interesting model that can tell you, it's called muscle injury model, that can actually, you can use the same nanoreporter, a dual nanoreporter that can monitor M1 and M2 macrophages in real time, right? You can see whether it's going to M1 to M2 or M2 to M1 in real time. So idea was we can use this to capture these macrophage phenotypic changes, which has never been shown before in disease models in real time. So this is where he used arginase 1. This is an M2 nanoreporter, where he inject the nanoreporter in the tumor microenvironment and see whether that shows M2 macrophages in the tumor or not. So this is the M2 nanoreporter. As you can see, this is the location of the tumor. As the time goes by, you see much higher activation that is happening. Free nanoreporter does show some responses, which were surprising because this is just a probe, but it does show responses, and you can get responses later on, not as early as the M2 nanoreporter, but we do see that a free probe also shows responses. Once you deplete the macrophages to show that this is macrophage specific responses, once we deplete the macrophage, there is no response that is shown in the tumor, whereas the previous control doesn't show anything. As you can see here, we can get much better responses in the M2 macrophages if you use a nanoreporter compared to a pre-probe. Here we used M1 nanoreporter, which is a natrium oxide nanoreporter, and we screened a variety of different types of drugs. In this case, drugs that can either deplete or reprogram M2 macrophages to an M1 phenotype, and our nanoparticle that I showed in the first study. You can see every drug has different types of or kinetics of responses that can be monitored, different locations that can go, different types of responses, different quantity of responses that can easily quantify using this nanoreporter. You can see here, if you use a nanoreporter, in this case our CSF1R inhibitor with the M1 nanoreporter, we get much better responses. So having the drug and the imaging probe in the same nanoparticle systems gives much higher sensitivity and selectivity of the responses that are shown here. And this is the tumor volume that I showed, which validates that, if you get higher responses or higher reprogramming, you get higher signal, which is M1 macrophage specific. And the final study, which is where we use this turpentine oil, which is a muscle injury model, where it leads to inflammation first and then resolution second. But once we inject this M1 and M2 nanoreporter, dual nanoreporter, we can monitor this activity inflammation early on. So there's an inflammation that is happening early on. Again, free probe shows some responses, but the nanoparticle system has much better, much quicker responses. And healthy mice, you don't really see much responses happening at that site. So this is where you have M1 nanoreporter that is showing the responses, and this is an M2 nanoreporter that is showing the responses. So initially, you have this muscle injury that leads to inflammation, which leads to M1 macrophages and nitric oxide release that can be monitored early on. And after a certain time, there is a resolution phase, which is a regeneration phase where you get much higher M2 macrophages, and that is monitored here. So in other words, we can monitor this reprogramming of M1 to M2 and M2 to M1 in real time, or higher M1 and M2 ratios in real time in disease models. All right, to just summarize, I hope I have convinced you that we have designed nanoreporters that, these are activatable nanoreporters that can allow us to monitor the immunotherapy responses, monitor the kinetics of responses, and how immune cell functions, what are the different drugs that can respond to those. And you can monitor all these kinetics of responses in real time, and can get an idea about how this can be used for monitoring which tumors are going to respond, which tumors are not going to respond. All right, with that, I would like to conclude and happy to answer any questions you may have.
Okay, thank you, Professor Kulkarni, for this wonderful talk. We'll take questions. I will ask audience to put all the questions in the chat box so that they'll be addressed. So, we'll answer the first question that is raised by Pragati Sharma. So she asked how T cells and macrophages identifies or recognizes cancer cells as cancer cells, which have CD47 receptor as a signal. So how immune cell differentiate normal and cancer cells?
That's a great question, and that's something that I would think is very critical for understanding how our immune system is capable of differentiating normal cells versus, or healthy cells versus cancer cells. Now, cancer cells have, obviously they have CD47 and other things as well, but cancer cell can also have, proteins that are expressed, right? Cancer cells generally are mutated, and that mutation leads to this presentation of antigen on their surface that can be recognized by T cells. Macrophage can generally figure out if they need to go and eat up the cells based on CD47 CYP5 interactions, and based on if they're, they consider that as a threat. So in other words, T cells have a different mechanism of recognizing tumor cells versus normal cells, whereas macrophage has a different way of recognizing self versus non-self. So innate and adaptive immune system have a different mechanisms of differentiating between self and non-self cells. Okay.
Okay. We have a next question from Sudeep. That is, what is the state of liposome-based delivery system for multiple drug systems, including monoclonal antibodies and ADCs?
Yeah, I mean, liposomal-based systems have been used for, mostly small molecule drugs. They generally can be conjugated with antibodies. Now, compared to antibody drug conjugates, which are a different class of molecules, the advantage of using liposomal system or system that are supramolecular systems is you can get much higher drug to antibody ratio, right? So if you have an antibody that is conjugated on the liposome or any other nanoparticle and the drug that is encapsulated or loaded in the nanoparticle, you get much higher drug to antibody ratio compared to antibody drug conjugates. So you can deliver more drug with lower number of antibodies to the tumor environment. Obviously, ADCs have their own advantages as well, but people have used liposomal systems as well to deliver antibody and small molecules, but liposomal systems get much better, antibody drug ratio compared to ADCs.
Yeah. So in line to that question, I would like to ask one more question. That is, let's say this ADCs, these are they're mostly being used, to have a target specific, site specific drug delivery. So how efficient this liposomal would be compared to, let's say ADCs, or you have liposomal assemblies along with the antibodies attached to,
Yeah, so one concern obviously is, the size of the molecule, right? The ADCs are relatively smaller size compared to nanoparticle system. The circulation, the stability, and the accumulation in the tumor, right? Generally, antibodies can go better, accumulate in the tumor, can circulate longer, so they can generally are more better in terms of getting into tumor microenvironment. But nanoparticle system can deliver more drug, right? And that's the thing, but they cannot really penetrate deep into the tumor as compared to smaller molecules. So ADCs have their own advantages, obviously, right? You can get, so for example, ADCs, you can conjugate drug that have picomolar toxicities, but which is what ADCs have been used for, delivering drugs that have picomolar efficacy, which you cannot use it to deliver using or as a free drug. Liposomal systems are very tricky because those molecules are very tricky to deliver using liposomal systems. These are a little bit larger molecules. So ADCs have been used for a very different purpose compared to liposomal systems, which have been used to deliver drugs that that are not as toxic. Size, stability, circulation, and then accumulation in the tumor are different for liposomal versus ADCs.
Okay. We have a next question from Sanjay. He asks, how did you model lipid bilayer? Can we study the drug transport as well using this model across the lipid membrane formation?
Yeah, that's a great question. I think Sudip can give more information about how they model the lipid bilayer system, but I can talk about the drug transport. Obviously, this membrane can be used for drug transport. Traditionally, has very widely been used as a surrogate for cell membrane. So that can be used to see drug interactions to the membrane and drug transport, right? How much of the drug passes through the cell membrane. So traditionally, the lipid bilayer system has been used to see the drug transport, drug interactions, mostly for drug discovery aspect, not so much for drug delivery. So yeah, I mean, it can be used to study drug transport using this model as well.
wait for some more time to have some more questions.
Maybe I'll start sharing so I can see.
Yeah,
Uh,
yeah,
okay.
would be fine.
Ashish, one quick question instead of typing, I just ask you, means
Yeah, absolutely. Yeah.
there is quite a confusion between LNPs and liposomes.
Yeah.
I am also very much confused and I don't know where to look for. Can you please tell us that how to, judge what is LNP and what is a liposome?
So LNP has, liposomes we mostly consist of either one or two lipids, different, not so much in terms of delivering, we use mostly delivering hydrophobic or hydrophilic drugs, right? These are liposomes. Liposomal systems are generally unilamellar, which generally has a hydrophobic shell and a hydrophilic core, right? That's simple system, a bilayer system is generally considered a liposome. And so you have a hydrophobic, and then you have a peg layer on the surface that acts as a stilt layer. So that's the liposome in a very broad context. Okay. Now LNPs have additional components. So you have this ionizable lipid, right, component. You have this cationic lipid component, and you have PEG component. Now these lipids have been used for very specific purpose. The purpose is to load the mRNA, right, which is differently charged molecules, so negatively charged molecule using the cationic lipid, right? And use ionizable lipid, which is serves another purpose of making sure that that mRNA escapes the endolysomal compartment, right? Because you want the mRNA to be in the cytosol that can be then translated. So as I said in one couple of slides, I have shown TM. You can clearly see the bilayer for liposomal system, right? You can see a clearly bilayer and aqueous layer. Now LNPs are tricky because they might have the small parts of lipids that are coated, that are coating your mRNA, right? It's protecting the mRNA in the system. Now, so it all depends on, and their sizes could be different. They're, obviously it's going to be spherical because that's the most optimal structure. But LNPs have these additional components that makes them load, anionic nucleic acids like mRNA. So you have to use ionizable lipid and cationic lipid to do that. So
we have done some work on mRNA and siRNA, and then we have seen that compartments and all. And so we were telling that this is liposomes only, means, okay, there are liposomes inside, there are compartments. But I was thinking there is what's wrong in calling them liposomes. Then after that, we understood people call them LNPs. And so somebody told us that, no, you should be calling them LNPs. And so basically liposomes are kind of a one class of LNP that can be, I think we can say.
that is true. That is true. So I would say LNP is a class of liposomes because, liposomes are very broad term, right? It could be multilamellar, unilamellar, could be different size, different shapes and things like that. So LNP could be considered part of vesicle or liposomal systems, but want to differentiate LNPs because they have a very specific composition, right? That your normal
ionizable lipid is the main thing in the composition.
Yeah.
If you have call it LNP, basically, that's another definition.
Yeah.
Thanks.
Yeah, I'll ask one question. So could you please discuss about the shelf life of this liposomes, or let's say LNPs? What are the factors, affects their stability?
That's a great question. I mean, we actually did the stability of this for over a month in 4 degrees, which is what we generally store those. And they're very stable, right? And this stability could be affected by, a lot of different factors, mostly, how we can maintain the structural integrity of the lipid bilayer system, right? A lot of times what happens that is water, dilution factors, water can interact and make this unstable. You can obviously put stabilizing factors, sugars and others that can make this liposomal system more stable during storage conditions, which people do when they're actually doing scale up and other things. But normally, you want to make sure that the lipid bilayer is stable. with the drug should not decompose or drug should not get degraded, when you are putting in the storage conditions. So there are several factors that can affect the stability. For example, you want to make sure this liposomal system maintain the colloidal stability. They should not just precipitate and settle down, right? They should not aggregate. If they get aggregated, then you generally lose it. So you have to use enough PEG layer on the surface or protective layer on the surface that they don't self-aggregate, things like that. So, it could be drug, lipid, could be size, could be, the interactions of the components with the water that can impact the aggregation as well. So.
Okay, yeah, since there are no more questions, yeah, again, I would like to thank Professor Kulkarni for enlightening us on several aspects of this nano cargos as well as drug delivery approaches. So yeah, once again, thank you very much. Now I will ask Dr. Seema to give a word of thanks.
Thank you so much.
Dr. Seema?
Yeah, hi.
Yeah.
Hi, Ashish.
Hi.
It's great to see you after ages. And I must say you look good.
Thank you.
And on behalf of everyone here at Prescience in Silico, let me first offer our congratulations to you on receiving the Langert Prize for Innovation and Entrepreneurial Excellence,
This is a huge
endowed by the American Institute of Chemical Engineers. What an incredible achievement. Congratulations, Ashish.
Thank you very much.
And with that, I would also like to extend our heartfelt thanks for your outstanding talk today. It was your insights on these nanoscale approaches for therapeutic immune modulation. It was really, really thought-provoking and really pertinent and invaluable to the current ongoing research in the field. I would like to say it was truly a pleasure to have you as an invited speaker today. And we're grateful for the time and effort that you made for joining us so early in the morning, considering East Coast time. So thank you once again. And we wish you continued success in your work.
Thank you so much, Seema. Again, thank you for invitations and all the great questions. I really enjoyed all the interactions and hope to catch up with all of you sometime in the future. So.
Absolutely.
Awesome.
Whenever you are in India, let us know. We will be we'd like to host you at Prescience. So please
Absolutely.
Yes.
Yeah, I would love to, for sure. Absolutely.
Yeah.
Good luck with Prescience. You're doing awesome work. I think, Sudeep has done a Herculean job of a lot of different things that he has done. So I hope you all, best wishes for your continued work. And yeah, it was really nice to interact with you and share our work. So.
Thank you.
Thank you, Ashish.
Take care. Bye.
Yeah,
Bye-bye.
bye-bye. Thank you, everyone, for joining. Bye.